Hello, Assalamualaikum, and good morning, everyone. This morning we have another breakfast at UMHealth series, and we have Dr. Lei Cheng as the first speaker, and Dr. Snellata for the second speaker. So first of all, I'm Dr. Naguib from Physiology Department, and I would like to introduce you Dr. Lei. Dr. Lei is a staff from Physiology Department, and this is a brief overview on her. She holds a BSc and a PhD from Imperial College London, and joined us about 6 years ago. And she primarily specialized on cellular and molecular physiology and immunophysiology. Her research, interest is on the function of proteins, in this case, the kinase, which is a protein that holds a great promise as a drug targets for the treatment of various diseases. Today, she will give us a talk on the function of this protein as a potential for it to be used for treating diseases. So as an academic staff of our faculty, so she also involved in various physiology teaching. So without any further delay, I would like to invite Dr. Lei to give the talk this morning. Yeah, over to you, Dr. Lei.
Thank you, Prof. Naguib. Thank you for the kind introduction. Let me share my screen for a moment. Okay, so I hope that you can see the screen. So good morning. This is a phylogenetic tree shows a human kinome. People are asking what is human kinome? A human kinome is a set of protein kinases in human genome, and it is comprises with 8 main groups differentiated by its conserved catalytic domain and based on the sequence similarity in the kinase domain. So if you look at the phylogenetics tree, we have the protein kinases that fall under category of tyrosine kinase, and we have tyrosine kinase light, and we have the STE 20, 11, and 7 related under one group. The casein kinase 1, the first protein kinase is being studied. Another group is the AGC, which is protein kinase A, G, and protein kinase C related under one group. The CAMK is where is the calcium ions or calmodulin dependent kinases. Another group is CMGC, which is for the CDKs, MAPK, GSK, CDK lights related. And also a small branch here is the RGC, the receptor guanylate cyclase. So in 2002, Manning et al. Reported that they have identified 518 protein kinases gene. Until today, the human genomes contain 530 genes, and it has been identified and coded. So what is actually a kinase? So kinase is a type of enzyme or protein that catalyze or accelerate this transfers of a phosphate group from the ATP on a specific substrate protein. This protein is being phosphorylated by the help of protein kinases. And this process is called phosphorylation. Phosphorylation can alter protein activity, localizations, and interaction. Just for your information from this diagram, the process of phosphorylation can be reversed by an enzyme called protein phosphatase. A protein phosphatase catalyzes the removal of this protein phosphate group from the phosphorylated protein, and this is known as dephosphorylation. And today I'm going to talk just about protein kinase, the phosphorylation. So kinases play a pivotal role in cellular signaling, where they act as molecular switches, turning celluloses on and off. Kinases regulate various metabolic pathways, such as glycogen synthase kinase 3, as I picked one of the example. The GSK3 inhibit glycogen synthesis by phosphorylate this glycogen synthase, or insulin receptors tyrosine kinase, which initiate the insulin cascade. Kinases also control cell division and differentiation. For example, cyclin kinase, cyclin-dependent kinases, the CDK1, 2, 6, and 4, are primary drivers of the cell cycle. Together with the cyclin, the CDKs and cyclin complexes regulate different phases of the cell cycle. Also, the kinases participate in the programmed cell death called apoptosis, and etc. So by using a BCR-able oncoprotein, BCR-able tyrosine kinase, represent a type of kinases in here. On the left side, the BCR-able, this cartoon, shows that it can bind an ATP in the kinase pocket. This is a kinase pocket. The BCR-able substrate is activated by the phosphorylation of one of its tyrosine residue. And where the BCR will transfer one of the phosphate group from the ATP to the tyrosine residue of its substrate. Then the phosphorylated substrate can activate the other downstream effectors molecule. A quick introduction about BCR-able protein kinase. It is a fusion oncoprotein result from a specific chromosomal translocation in a chronic myeloid leukemia. It activates signaling pathway crucial for CML progression and represent a target for targeted chemotherapy, which is imatinib. It is a kinase inhibitor and used to block the phosphorylation. On the right side of this figure show when imatinib occupy the kinase pocket, which is also the ATP binding pocket. The BCR-able action is being inhibited by the imatinib, prevent the phosphorylation of its substrate, and subsequently block the leukemogenic effect of the Philadelphia chromosome. Moving forward, we understand that kinase can be inhibited by having a small molecule or inhibitor or drug that can sit in the ATP pocket, which then inhibits its phosphorylation. From the past, most of the kinase inhibitors that have been developed for use in clinic act by blocking the ATP binding site. Now here I share a figure showing different types of kinase inhibitors. The type 1 and the type 2 targets the ATP sites. The difference between these two types is that the type 1 targets the ATP site when the kinase is inactive and is where the DFG motif conformation of this activation loop is phased into the pocket. While the type 2 target the ATP binding site where the inactive kinase conformation when the DFG out motif is facing out the pocket. So bear in mind, kinases can be also in the form of active or inactive, open and closed. There is a growing interest in identifying small molecules or compounds or inhibitors of drug that target kinase activity and function without interfering with the conserved features of the ATP binding site. So the type 3 kinase inhibitors have been identified to target site adjacent to the ATP binding site in the catalytic domain, while the type 4 kinase inhibitors target the region outside the catalytic domain, which is distant from the ATP binding site. And this site we call the allosteric sites. Further, the combinations of targeting unique allosteric site outside the catalytic domain with the ATP-targeted compound has produced a novel bivalent type 5 kinase inhibitor. And finally, the type 6 kinase inhibitor is an irreversible compound that form selective covalent interaction with the key amino acid involved in the kinase function. This figure shows the kinase drug discovery. The first protein kinase inhibitors was Fasadu, which was approved in Japan in 1995 for cerebral vasospasm, followed by sirolimus, the first allosteric kinase inhibitors, which was approved in 1999 for the use of in combination with cyclosporine for the prevention of organ rejection in patients receiving renal transplant. Then in 2001, the first economical successful protein kinase inhibitor, imatinib, or its brand name Clavich, was approved for CML and later for gastrointestinal stromal tumors, following on the emergence of kinase inhibitors approved for a number of kinase targets in different diseases. Here is another timeline depicting important events in the development and approval of kinase inhibitors over the 20 years since imatinib was approved for treatment of CML in 2001. Next, I'm going to talk about kinase that I am interested in. It is a leucine-rich repeat kinase 2 known as LAC2. The LAC2 gene encodes a large multi-domain protein linked to the pathogenesis of Parkinson's disease. The LAC2 is a 288 kilodalton polypeptide containing 7 structural domains, which is the armadillo repeat, anchoric repeat, leucine-rich repeat, a typical Rocco family GTPase domain, and 2 tandem fold C-terminal of ROC, which is the core A and core B, fall under the category of COR. Of course, a catalytic kinase domain, and WD-40, a beta propolator. So LAC2 is a type of serine threonine kinases. The RAP10 and its other RAP protein have been identified as physiological substrate for LAC2. And LAC2 phosphorylate RAP10 protein at the conserved threonine serine motif. And this site become the biomarker site of LAC2 kinase activity. And it is important to understand the role of the protein before it is selected to be drug. So LAC2 involved in several cellular processes. LAC2 can bind directly to the microtubules and interacts and its interaction seems to influence microtubules stability. LAC2s play a significant role in mitochondrial functions. It interacts with protein involved in mitochondrial fission and fusion, affecting the shape and also the size of mitochondria. LAC2 is involved in regulating calcium level in mitochondria, which is crucial for various cellular functions. Research indicates that LAC2 can inhibit formation of autophagosome, the structures involved in cellular waste disposal. This inhibition might contribute to accumulation of the toxic protein seen in Parkinson's disease. Lysosomes are membrane-enclosed organelles that contain an array of enzymes capable of breaking down all types of biological polymers. So LAC2s can influence the structures and functions of lysosomal membrane. It has also been implicated in the formation of lysosomal tubules, which may be involved in release content from lysosome. Studies have shown LAC2 involved in the movement and recycling of synaptic vesicles and impact the process of neurotransmitter release. The disruption of this process can lead to impact neuronal signal. So I have shown the role of LAC2 protein kinase. involved in our body, and why it is important, why LAC2 is important. While the underlying cause of Parkinson's disease is unknown, LAC2 mutation are the most common genetic cause of Parkinson's disease, accounting for 5% of familial cases and 1% of sporadic cases. The most common pathogenic mutation is the G2019S, which is the glycine at the location of 2019 amino acid changed to become serine. And it is common in the white patient and is present up to 20% of Ashkenazi Jews and 40% is seen in the North African Arab patient. However, it is almost completely absent in Asian. On the other hand, Asian-specific variants are found exclusively in selected population. For example, the G2385R at the WD domain and R1628P at the core A domain. These variants are the most common reported genetic risk factor in Chinese individuals. The G2385R is also common in Japanese and Korean individuals. And these variants are absent or rare in white people and in other Asian groups such as Indian people. Today, there are about 215 variants identified in people with PD. And 215 variants in LAC2 has been identified in PD in people with PD. And these figures is only highlighting the location of 45 variants that have been reported to promote LAC2 kinase activity at least 1.5-fold in cellular overexpression assay of RAP phosphorylation. So the substitutes of the serine for glycine at the position of a 2019 amino acid in the human LAC2 protein is the common mutation. And this mutation happened at the kinase domain. And this variant is a pathogenic mutation, and it has shown that it enhances the LAC2 kinase activity moderately. Studies have shown increase of the LAC2 kinase activity cause mitochondrial dysfunction, impact autophagy, which is a process by which a cell breaks down and destroys all damaged abnormal protein in the cytoplasm, synaptic dysfunction, inflammation, decreased lysosomal degradative capacity, alter lysosome structures. And these may contribute to the development of PD, which is still under investigation. Therefore, it is crucial to inhibit the LAC2 mediated RAP10 phosphorylation. Knowing the importance of drugging the LAC2, especially cells with G2019S mutation, a project was initiated to study novel small molecule in inhibiting LAC2 kinase activity in LAC2 G2019S cell model. A series of a small molecule was synthesized. This series of small molecule has the same backbone of MLI2, which has been proven to be potent and selective as a LAC2 kinase activity, but it has shown to have on-target side effect on lung and kidney. 17 novel small molecules were screened using the in vitro cell-based assay to assess the LAC2 kinase activity. In addition to the protein-ligand interaction, it was analyzed at the atomic level by using the method of molecular docking. So 1 out of 17 small molecule was found to significantly inhibit the phosphorylation of RAP10 at threonine 73 in the wild-type LAC2 cells without affecting the phosphorylation of LAC2 serine 935. And this suggests that this compound, small molecule, is a type of type 2 LAC2 kinase activity. There are also 3 small molecules in this series resulted in decrease in both the total LAC2 protein levels and also the LAC2 serine 3,5. And we suggest that maybe this small molecule may possibly be a LAC2 inhibitors that induce destabilization of the protein. This study is conducted by Anis Nadira, our Masters of Medical Science student. So following on, these are the strategy by knowing the LAC2 protein functions and the domain. These are some of the strategy that we can drug the kinase. One of it at the armadillo repeat domain, which is found to have the binding capacity with the substrate, which is the RAD10. The inhibitors can be generated to bind on this site. At the anchoring domain, repeat domain, can design inhibitors that bind on the anchoring as the allosteric type. While the leucine-rich repeat has the binding sites for the PROTAC. I'll talk a little bit about PROTAC later on, which is help to degrade the whole total LAC2. At the ROCO domain, an inhibitors that bind allosterically can be designed. Also, the non-atypical GTPase binding, where this actually cause the phosphorylation, help the phosphorylation, and inhibitors can also be designed at this site. Nonetheless, the kinase domain, where different types of inhibitors like 1, 2, and 3 targeting the kinase domain. And lastly, the WD domain, which is function as helping the 2 LAC2 to form a dimer, and a strategy to design the modulators to prevent the dimerizations of interactions. So I will talk about a little bit about PROTACs, which is an emergence of this proteolysis targeting chimera, short name called PROTAC. It resulted in the discovery of the degraders. So PROTACs are an innovative molecule that revolutionize this drug development by eliminate the target protein. The heterobifunctional degraders can induce the target protein to degrade by recruiting E3 ubiquitin ligase in the proximity to the target protein, resulted in the polyubiquitination and subsequently degrade the target protein by the proteasome. And here these figures shows an example of fast, potent, cooperative, selective oral bioavailability and blood-brain barrier penetrance PROTAC degraders of LAC2, which is XL01126. Therefore, this is my last slide, and I would like to acknowledge the funders for the projects, the studies that have been conducted by FRGS MOHI and UMRG grant. These are the collaborators that have been supported me throughout these past few years, and definitely the group from the UMPD teams are engaging, and I'm happy with collaborating with, continue with this collaboration. And thank you very much.
Okay, thank you, Dr. Lee, for a very nice presentation. So I would like to open any questions from the participants. There is one question in the chat box. From the experience and knowledge, which site is the best to target to inhibit kinase activities effectively among the 6 types?
If you look at the figures that I showed, there's 6 different types of kinase inhibitors. Definitely people moving away from type 1 kinase inhibitors. It is because of the conserved domain of the kinase sequence. So the kinase sequence are conserved throughout these whole kinases. So if we design an inhibitor targeted on the conserved domain, it may have off-target effect. So therefore, the generation of the type 2 kinase inhibitors may be another options. People moving away also from type 2 now moving towards the third and the fourth, which is the allosteric site, which is much more specific site that doesn't cause any off-target effects. So each of these type of inhibitors has its has its pro and cons, and definitely the type 2, 3, 4, 5, and 6 is way better than the type 1, which is the traditional way of targeting the ATP binding site, which is the conserved domain.
Okay. Another question is, I would like to ask, currently for Parkinson's disease, what is the latest treatment, and how does your LRRK2 inhibitor is more potent, or you presume that it's going to be a better drugs than the others?
At the moment, in clinics, the drugs or the medications that given is to help the patients in terms of their dopamine substitutes medications. In terms of LRRK2 drugs, it is still in clinical trials. So, for example, that previously the one of the very potent type 1 kinase inhibitors, which is MLI2, has been studied in the clinical trial, but however, it didn't pass the clinical trial because it caused some side effect on the kidney and lungs of the non-primates animals. Therefore, there's the need to improve this molecule. For the LRRK2 degraders, as I shared just now, PROTAC, still in the development, entering the clinical trials, which has helped to remove the total LRRK2 proteins. And we are hoping that the different types of LRRK2 inhibitors can be, as I shown in the last two slides, that there are many different ways of targeting these kinase, drugging these kinase at different domain. We are hoping that these inhibitors can help to, remove the LRRK2 proteins from patients who has high activities of LRRK2 kinase, or patients or inhibitors that actually can help to reduce the LRRK2 kinase activity. Therefore, it can actually suppress all the dysregulations cell processes in the patient.
Okay, there is a question from Prof. Tengkusara. At this stage, could you outline what needs to be done before we can see a clinical use for Parkinson's?
If I understand the question carefully, means that outline the inhibitors, is it? I think, I'm sorry, maybe I interpret the question as outline these inhibitors before it is used clinically. Stages that we need to go.
We need to go through. Yeah.
Okay, so the development, I believe, is the development of the inhibitors. So there are many researchers has been working on different types of small molecules inhibitors, looking at the different site. For example, PROTAC, or type 1, type 2, or type 3 inhibitors. Some working on the cells models, and now we're moving towards these working on the biomarkers, means that getting patients blood cells or skin fibroblast cells, and we treat it with these molecules and see whether it can help to reduce the LRRK2 kinase activity. Then later on, once it has passed this on, we're working on the different types of animal models. Then later on, to hopefully they can enter the clinical trials for the humans. I hope I answer your question, Prof. Sarah.
Okay. Okay, if there is no more questions, I would like to thank Dr. Lee for a very interactive sessions. And without any further delay, I would hand this session to Dr. Intan, who will chair the next session. Thank you so much.
Thank you.